Good morning, everyone. So today we're getting started on chapter 6. And while I was prepping for this chapter, I kind of took a little bit of a step back and looked at our schedule for the next little while. And if we keep moving through stuff at the pace we've been going, we might be done far in advance of when we should be. So if you've been following along, you might realize that we technically have almost 2 weeks to go through all of this. But I only have 40 slides, so we're going to see how that goes. But at least we know that we'll have lots of time because the second midterm, so our next one that's coming up on November 2nd, that only covers up till the end of chapter 6. So we're actually going to start chapter 8, which is the next one, a little bit before that midterm starts, but I won't test on it. So we'll just have a little bit more time to sit with the chapter 6 material before the midterm, and then we'll kind of keep going, because I think I put more sort of wibbly wobbly room in the middle and less at the end, so we'll probably even out towards the end of the semester. But yeah, so that's sort of a nice segue into reminding you that we have a midterm coming up. So because we're now back to in-person, we're going to be doing the midterm in this room, and I'll be having it just sort of in the class as per normal, which is going to be kind of weird. It's been almost 2 years since I've done an in-person midterm, but yeah, shouldn't be anything fancy. Same basic setup, some multiple choice, some short answer questions. So the same basic layout. I'll probably do a couple fewer multiple choice questions. Just I find when you're doing them in person and having to sort of flip through pages, the timing's a little bit different, but I'll post the exact details on our Moodle when I have it all figured out. Okay, I think that was it for housekeeping stuff. So we're going to move on, and we're talking about, well, I suppose the whole chapter is talking about motivation and regulating our internal states. So we're going to spend a lot of time talking about homeostasis and things that sort of drive us to different kinds of internal type behaviors. So we can start off with our standard definition that motivation is a set of factors that initiate, sustain, and direct our behaviors. So if we think of motivation, that's the process of setting something in motion. So if we think about it as our internal state of being set into motion. And so this was kind of proposed because we weren't able to explain behavior entirely in terms of just external stimuli. So if you remember back to our history section where we were talking about our behaviorists, and especially our radical behaviorists who loved the idea of all of our behavior being driven by external environmental forces. But when you actually get into sort of human behavior in particular, but most behavior in general, we start seeing that it isn't just the environment that explains things. We have to have something else to go along with it. At least if you're not still a behaviorist, they would still disagree with this entire chapter. But it is important to remember that this is sort of an invented concept. This is us trying to quantify or discuss those other factors that aren't just our environment. So we don't have something that is like we can look at it and directly measure something as motivation. There isn't a part of the brain that lights up for that. There isn't sort of one specific thing that is motivation, but it's our way of talking about all of the other things that we're not quite sure of. So just keep in mind that it was sort of an invented concept in order to allow us to talk about what we need to talk about in this chapter. And so we can also talk about instinct, which is almost like the opposite of motivation, I guess, depending on your framing. But instinct would be the idea of automatic behavior, things that happen without any kind of learning, things that aren't able to be changed through the process of learning, and might even be complex, that occur in all members of a species. So instinct should be something that, from this definition, is probably genetically hardwired. Oh, yep.
I have a question about the unmodifiable part of it. So I took a class a while ago about abnormal psych, and we talked about phobias and how some phobias might be instinct-driven, like fear of spiders or fear of snakes, because they can kill you. I'm just wondering if exposure therapy can kind of modify the instinct that's there, or if it's just...
It could lessen the behavior that's responding to the instinct, but like the instinct itself is sort of the driving force, the almost like motivation, but we're going to kind of talk about motivation more as, well, I guess, a subset of. But it's sort of a biological based thing that's naturally occurring that doesn't typically change. We can sort of modulate our response to it, but especially when you see instinct in the literature, like the proper use of it is going to be for things like migration, or if you've seen like fixed action patterns where they have very rigorous steps of behaviors that occur together. That's usually what they're referring to. And so that kind of gives me a good crossover. So if we're saying that this is something that's automatically happening, it's probably in our genetic code. And so I kind of hinted at fixed action patterns. It's one of my favorite ones to look at. So if you've ever seen an explanation of fixed action patterns, you've probably seen egg rolling behavior in geese. And so the idea is that while they're sitting on a nest after having laid eggs, they have this sort of innate instinctual behavior to return their eggs to the nest. So if they see something that looks like an egg, they'll initiate this rolling behavior to sort of stand up, stretch their neck out, and then scoot the egg back underneath them. And this fixed action pattern happens in a very strict, rigid sequence from beginning to end, even to the point that if you went out and picked up the object that they were rolling, like as soon as they touched it or as soon as they initiated it, even if they just stood up to start it, they would continue the full action, bringing nothing back to the nest to sit on. And you can actually play with stuff like this by sort of messing with which stimulus you're using. So maybe you can make it more extreme. It doesn't even have to be egg-shaped. You can do it with things that are cylinders. Researchers did lots and lots of tests to see what they would do this with, and they would move things like pop cans and cubes and whatever they could put in front of the goose could potentially initiate this pattern. So that's a very rigid and harsh predictable version of an instinct, but we also see instinct in things like migration or parental behaviors in different animals. So we have some of these sort of built-in behaviors that allow us to do things that are important for our survival. So if we think of migration, this is when mostly birds, but we also see it in aquatic creatures as well, where they'll return to the same place year after year to winter there and then return somewhere else to spend the rest of the year. And they seem to follow pretty fixed pathways. They seem to do it at a similar time of year and all of that. And because that isn't necessarily learned, we could use something like sea turtles as a great example of this, because they're hatched on a beach and sort of instinctively know where to go, even though there are no adults around for them to learn from. So that's some of our evidence for instinctual behaviors. And so these are all just ways of describing what might be pushing us to do different things. And it's going to tie us into this concept of drive theory. And so drive theory is based on the idea that our body tries to maintain the condition of homeostasis. And we've talked about homeostasis before for lots of different things at this point, but it's just the state in which any particular system within the body is in a balance or equilibrium. And as I always say, this could be something as simple as heart rate or body temperature, where we have sort of an optimal range, where if our heart rate is too low, then we have trouble getting blood everywhere, but if it's too high, it's not healthy and it causes problems in other places. If our temperature is too high or too low, then our proteins and our cells won't function correctly. So we have these optimal ranges in which our body wants to function, and that's what homeostasis is referring to. So drive theory is based on the idea that if we are pushed out of that balance, our body is going to strive to bring us back into balance. So drive, they've described it here as an aroused condition, which again just means that we have sort of a high energy state where we're going to be trying to do something to fix that issue. So when we have a drive, we're pushing to get back to balance, to get back to homeostasis. And you'll usually see drive theory and incentive theory talked about together, and that's because they were originally considered sort of one and the same. It was drive and incentives theory all at once, but then they kind of separated out. And so now we look at sort of drive theory as the internal component, and then incentive theory is that external component. So incentive theory focuses on our motivation from external stimuli, things in our environment, not just our internal drives. We can also talk about arousal theory. This is another weird one. I haven't seen too much of this. And this is, again, talking about sort of level of activity in our nervous system, where different people have different preferred levels of arousal. So maybe people really like being constantly stimulated by their environment. Maybe they're thrill seekers. Maybe they really want adrenaline and go all the time. So they would behave in a way that keeps them at that preferred level of stimulation. Other people might prefer to have very low levels. They'd rather be somewhere quiet, and they'd rather have nothing going on. They'd rather have very low levels of stimulation. And so they'll choose to do actions that kind of keep them in that level. And so all of these, again, we run into this issue where none of them are necessarily sort of right or wrong. They might all kind of work in conjunction with one another, but different theories are used depending on what you're sort of shaping your ideas around. And so the early version of drive theory was really focused on sort of not being centered around the brain and not being involved at sort of higher levels of cognition. It was more of your body is doing what it needs to do to stay in balance. So your heart would make adjustments, or your stomach would make adjustments, or whatever. But that was sort of the old version of drive theory. And as we get more and more evidence, and as we start sort of including more and more theories, we've shifted to emphasize drives as more of a state at the level of the brain. So we have centers that monitor and register what's going on in our body. We would have sort of something monitoring our internal temperature, and if it's too hot, then we're going to initiate behaviors that will reduce that. Things like sweating, or maybe taking off an outer layer in order to cool down. So we can have sort of bodily responses, but also behavioral responses. So as time goes on, we incorporate more of these things, and it gets a little bit more complicated. But if we wanted to talk about how this drive theory works, looking at a particular concept, we're going to look at temperature. So this is a simple homeostatic drive. We're mostly going to frame our understanding in terms of the more simplified systems, because this can get very complex and they get a little bit intertwined as time goes on. But this is going to be a good place to start, because it's going to allow us to walk through drive theory, not just with an abstract concept, but with a specific example. And so we're going to have some terms that are useful here. If we wanted to talk about our equilibrium, that homeostatic sort of happy point, we can call that a set point. That's that sort of balance where our system is trying to return to. And you usually see set point, at least from my experience in teaching psych, we usually see set point when it refers to weight. So if you would look at sort of a body mass fat to muscle ratio, individuals tend to have a particular set point where even if they diet or exercise, their body tends to gravitate to the same weight. And that's just because that's where the body is at equilibrium. So it takes a little bit more effort to move that set point. But that concept doesn't just apply to weight. It can also apply to literally any aspect of homeostasis. So any point that we're talking about can be a set point depending on what system we're referring to. So here, if we're talking about temperature, our set point would be our optimal range of body temperature to function. So we have an optimal range somewhere around, I think it's like 36 to 37 degrees Celsius. Some people will run a little bit warmer, some people will run a little bit cooler, depending, but we all sit right around there. And so in different species, we have different kinds of temperature regulation. So we will briefly mention the fact that we have ectothermic animals, Ecto meaning external, thermic just for temperature. So these would be snakes and lizards, and so what they do is they rely on their environment in order to balance their temperature. So if you've ever seen lizards sunbathing on a rock, they're raising their internal temperature, probably bringing up their temperature to allow them to digest food or do anything that requires sort of energy in the body. Then they'll move and go somewhere cooler if they want to bring their body temperature back down again. So they're sort of relying on these external stimuli, external temperatures in order to regulate their own internal temperature. And then for the rest of us, we are endothermic, endo meaning within, so we rely on internal processes to maintain our body temperature. And we actually have a part of our brain, so the preoptic area, which it's in our sort of mammalian hypothalamus. There's something sort of equivalent in other species as well, but it contains separate warmth-sensitive and cold-sensitive cells. So that's that part that monitors if we are too hot or too cold or just right. So we have a system in place, kind of like a thermostat, to tell our body if we need more or less heat to stay at our nice set point. And I do find it kind of interesting to note here that snakes and lizards are ectothermic, but birds and mammals are endothermic. But birds are more closely related to reptiles than to mammals. So I wonder if we've evolved endothermic sort of bodies twice, or if we were originally endothermic and then ectothermia evolved the once. As someone with a little bit of an evolution background, that makes me curious, and now I kind of want to look it up. But if any of you know, you can let me know. All right. Oh, okay, I didn't have another slide to talk about that, so I always worry about going off on my tangents without sort of advancing the slides, and then I realized that I intended to tangent here, so that's fine. Okay, so we left off before my evolution tangent, talking about, okay, so the part of the brain, the preoptic area, is where we have that thermostat, the warm versus cold sensitive. And I already kind of mentioned this, but we have different activities or different bodily responses that can be triggered if we are too warm or too cold. So if our cold sensitive cells are going off, it's telling us that our body temperature is too low and we should do things that raise our core temperature. So this would be things like shivering or increasing blood flow to, say, the core part of our body. In extremes, like in the case of, say, hypothermia, our body can actually deflect blood from our extremities, so condense everything into the middle of our body in order to keep all of our vital organs functioning at sort of the risk of causing damage to our extremities. But these are sort of intentional things that are happening to try and maintain that core temperature at that optimal level or that set point that we need to keep all of those organs functioning. Mm-hmm.
I think this is kind of just a comment. I think it was Dr. Borico that told me about this, but apparently a brain freeze is because your stomach is cold and your brain's like, let's send blood down there, and then there's not enough blood left in the brain to keep the brain warm.
Interesting. I've never heard that explanation, but I kind of like it. It does also make sense because our digestion's happening through enzymes and they change shape based on temperature. So if they're too hot or too cold, they can't function optimally. So it would make sense to have some kind of mechanism to get them back to where they should be. Interesting. All right, and we can also have, like, I guess, one last thing to mention on this idea of preparing different parts of our body or reacting in particular ways to being too hot or too cold. We can also have longer term responses. So if we are continually too cold, that can trigger us to start storing fat reserves to help us stay warm. Or if you're too warm, you might go the other way and try and burn off some of that. But that seems to work a little bit more for, say, species that rely on environmental cues for, say, hibernation or seasonal regulations like that, which is far more complex than what we're covering today. The second of our simple homeostatic drives is to look at thirst. And so you're going to find that we're kind of working our way up in complexity. So we had sort of a single slide for temperature. That was pretty straightforward. We have our sort of mental thermostat, and we can adjust our behavior and also our internal actions, whether we need to be warmer or cooler. And now for thirst, we're going to start talking about sort of different kinds of thirst and then also the multiple organs that are involved in this process. So our definition for thirst is dry mouth and throat that influence when to drink, but it doesn't necessarily influence how much to drink. So there are other aspects involved in determining how much, but whenever your mouth and throat are dry, that gives you that feeling of thirstiness, that desire to go get a drink. And so we have two different kinds of thirst. We can have hypovolemic thirst, and this is thirst occurring when our blood volume drops due to loss of extracellular water. So this is basically saying that we don't have a lot of free-flowing water in our bodies. Our blood volume is low, so everything is kind of lacking in water. So we're thirsty because everything in our body needs that water. But we can also have osmotic thirst, which is a condition that occurs when the fluid content inside the cells decreases. So instead of just the area around the cells and sort of all of that free-floating fluid within our body, this is everything inside the cells. And again, because cells need to be sort of at that happy middle point, they want to be at a set point for pretty much everything, not having enough water around can start causing problems too. And so these two kinds of thirst often occur together, so it kind of makes sense that if you don't have enough water outside the cells, you probably also don't have enough water inside the cells. But you can sometimes get osmotic thirst independently. So it can happen alone, specifically when you have, say, dissolved substances in the fluid around the cells that kind of pull the water out of the cells. So if you've ever heard about sort of freshwater fish versus saltwater fish, if you put a freshwater fish in salt water, all of the water will sort of osmose across the cells and cause problems because the water is going to move from areas of high concentration to low concentration, trying to balance out. Kind of like when we were talking about those ions moving across the cell membrane. Same idea here, but we're just looking at the concentration of water molecules. So if we add a lot of dissolved substances outside of our cells, then the concentration of water to whatever other things are around the cells is a lot lower than the water inside the cells, and so the water will leave, leaving our cells dehydrated. So that's when we can end up with this osmotic thirst.
So you said the osmotic could happen independently, but can the hypovolemic happen independently or always with the other one?
Always with the other one. Yeah, there's no really good way to build up, because like you'd have to have a really high concentration of stuff inside the cells, higher than outside, in order for the water to want to come in, but that can't really happen. And technically, if you did get it to happen, you would probably cause the cells to rupture, which is why you don't take saltwater fish and put them in fresh water. But yeah, so you don't ever see that happening. It's either all together or just osmotic if someone is still alive. All right. And then so for our structures involved, we're going to have a couple here, and they really, really like their acronyms. As you probably figured out from the way that I like to word my questions, I will usually give you both the full name and an acronym in a question, just because acronyms have almost no meaning to me. I can't keep the letters in the right order, so I'm going to give you the full thing and the acronym. So if you want to just learn the acronym or just the full name, either is fine. But yes, to save some space on the slides, we have both used and then just the acronyms. So hopefully I'll keep it straight. But we have our subfornical organ, or SFO. And this is a brain structure that's adjacent to our third ventricle. So if you remember, our ventricles are those parts of the brain that are kind of open and full of fluid. So that SFO is going to sense and regulate the internal water balance with our organum vaculosum lamina terminalis, or OVLT. So they're going to work in pairs there. So the SFO and OVLT are going to work together. They're both right around that third ventricle, and they're going to partner up to figure out what's going on inside the cell, or inside the body in general. So our third ventricle is just sort of one of the places that has lots of our spinal fluid. And because water osmoses or moves across membranes pretty freely, if we can measure the water levels at one part of the body, we can get a pretty good image of what's going on in the rest of the body. Okay. So these two different kinds of organs are what are called circumventricular organs, which is a fancy way of saying that they're outside of the blood-brain barrier, and they come in direct contact with sort of our blood circulation. So if we remember that our brain, we wanted to make sure that things from our blood can't just freely diffuse across into the brain, because there are things that might be circulating that could be harmful, that could damage our brain, and we want to maintain as much control as to what's coming in and out of the brain as possible. So we have that blood-brain barrier, sort of a coating around our brain, and anything that's coming to the brain from our blood needs to transfer across that membrane. So this is just saying that these two organs are outside of that blood-brain barrier, and they can directly access our blood in circulation, so they give us a more accurate representation of what's going on. And so some of the neurons within those two areas are what we call osmoreceptors. And if we break it down, osmo just meaning water. So if we were talking about osmosis, the process of water diffusing from an area of high concentration to low concentration, osmo is referring to the fact that it's water movement. So here, osmoreceptors, they're going to fire more as the volume decreases due to fluid loss. So they're receptors that pick up on the level of water. So as that volume starts decreasing, we're going to get more and more signals from these receptors that tell our brain that the levels of water are going down. So at first they don't fire too much because we're at a pretty good place, and then as it drops, they're going to fire more and more and more. All right, so our next location, or our next structure, is the median preoptic nucleus, or MNPO. And this is also adjacent to our third ventricle, so they're all in that same part of the brain. And it's going to integrate information from both our SFO and OVLT. So basically taking information that's coming in from both of those other cells. What we're caring about mostly here are those osmoreceptor signals and sticking them together. Okay, sorry, I just wanted to double check my notes here. So when we're looking at hypovolemic thirst, so that is our first kind of thirst. That's our sort of everywhere in the body thirst, as opposed to our osmotic thirst, which is inside the cells. We're often getting sort of our initial signal coming from baroreceptors in the arteries of the heart. And so they're basically just going to be in our heart, and then they're going to register the volume of blood in the heart, sort of the pressure, I guess, is the best way to put it. If you think of a barometer helping with weather, that's measuring the atmospheric pressure. So baroreceptors are cells that are going to signal pressure within our body, specifically in the heart. And so then this is going to send its information up to that SFO and OVLT, which will then sort of mix everything together in our median preoptic nucleus. So lots and lots of sort of connections here and lots of different ways to monitor this. It's almost as though we have sort of as though it's very essential that we maintain the correct levels of blood volume in our body, the correct levels of sort of water for us to continue functioning. All right. We can also have some information sent using an angiotensin 2, which is a hormone, and it's basically going to tell our brain about a drop of blood volume. So this is what's telling our receptors in the SFO and OVLT, and this is coming from the kidneys. So again, multiple places are going to be sending signals up to our brain through those three objects or three interacting structures. So here, when our kidneys, which are responsible for sort of filtering stuff out of our blood and then also a convenient place to notice where our water levels are, when it notices that the volume of our blood is a lot lower, it's going to release a hormone called renin, and that's going to cause increase of angiotensin 2. So it's kind of a little bit of a hop for there. But that's how we go from that part of the body to let our brain know that our volume is low. And it's going to take a little while. So the other ones which were giving us a little bit more of a direct signal, so if we're looking at receptors, then we're going to be getting that neuronal signal. But here we're talking about hormones, and specifically a hormone that triggers the production of another hormone that lets our brain know. So in this case, it's going to take a lot longer for that signal to reach the brain. So this is why it might take you a while to realize that you're thirsty, especially if you're relying on this particular pathway. But it's going to take 10 to 20 minutes for your body to actually pick up on that. And that's why it can take so long to realize that you have fixed the problem. So you might end up over drinking water. You might still feel thirsty for a while, even though you've drank a ton of water, because it's going to take a long time for these signals to turn off. So it takes, about 10 to 20 minutes for that notification to turn off once your body figures out that everything's back to normal. And telling it to turn off would be an example of a signal of satiety, which is basically that you have satisfied that appetite, that you are no longer thirsty. So you have satiated that drive that was occurring in the background. Okay. And then just if you're curious about any of the locations of those sort of sections that we were talking about, there's a diagram here. Again, as you know, I don't love testing on visuals or locations, and we all just sort of care that they're all in that same area around the third ventricle. That is sufficient for us. But if you are a visual diagram type person, there's a diagram for that. Okay. So, as I said, we're working our way up through more and more complex drives. So we started with temperature, we had our one slide, and then we went over to thirst, and we had quite a few more slides. And now we're going to look at hunger. And we have enough content with hunger that we're probably not even going to finish it today. And that's perfectly fine. As I said, we have lots of time. But this is us sort of having worked up to an even more complex drive. And it's kind of interesting. I also teach an intro psych class where we've just done our sensation and perception chapter, which you'll remember from 104. And you break down and go through all of our different senses and how those work. And in that class, I spent maybe 5 or 6 slides on taste, and then we moved on. But here we're going to spend a little bit more and actually delve into it and understand what's going on at a bodily level, what's going on in our brain that helps us sort of register when we're hungry, when we're full, and everything in between. And so we can start off, again, kind of like we did with temperature when we were looking at ectothermic versus endothermic. We can also look at different preferences for food types. So you've probably already heard about herbivores and carnivores and omnivores. Herbivores mostly eating plants, carnivores eating meat, and then omnivores that are somewhere in the middle. And I found it really interesting as I got into my research with birds. I always assumed that, birds eat seeds, that's it. Birds are technically omnivores. I've seen quite a few instances where if you have, say, a roadkill deer on the side of the road, black-capped chickadees will sort of pick at its skin in order to get at the layer of fat because it's a really good source of, well, fat and energy and nutrition. So there's these pictures of these tiny little, say 12-gram black-capped chickadees who are standing on top of a deer, and they're just, picking at its skin and eating its fat. So that kind of ruined my idea of what category birds fell into. And since getting my own birds, I realized that they really do like meat when they get the chance, whether they should or not. So I think most birds fall under that omnivore category. So just a little random fact that I've learned fairly recently. Back to the actual content for today though. When we're looking at hunger, this is going to be quite a bit more involved than when we were looking at thirst. So when we were looking at water, it was basically, there's a decent level, there's a certain range of water, a certain concentration that we want within our body. But with eating, with hunger and food, that set point is going to be pretty different. And we already kind of mentioned that I've always seen set point referring to, say, body weight, sort of that ratio of fat to muscle and your overall mass. But we end up having sort of lots of different things going on with hunger. So when we were looking at temperature regulation and thirst, we were looking at sort of one moving variable. So temperature is just the amount of energy in our cells, and thirst was looking at the amount of water, but hunger is going to be dealing with lots of other things. We don't have just one kind of nutrient. We're not just looking for energy. We're not just looking for, say, glucose. We're also looking for other nutrients, other things that our body needs to survive. And that's where the complexity of this starts coming in. And so we can have different kinds of sort of nutrients might be associated with different primary tastes. So we'll talk a little bit about our 5 primary tastes. You might have only heard about 4 of these. So the standard 4 are sweet, sour, bitter, and salty. But a lot more recently, we've seen this inclusion of a 5th one, which is umami. And even our sort of intro texts are lagging a little bit. They sort of add umami as an afterthought. But it's really having a lot of support as a 5th distinct category. And it corresponds to sort of like a savory flavor. And so I'm very excited to actually have it discussed here because all the other textbooks I've used have mentioned it but never actually taught us about it. So we're going to be able to talk just a little bit about all of those. Okay, and I just wanted to double check where I was going with this. So when we're talking about our receptors for taste, if we want to know how we're getting that signal from our mouth to our brain to tell us what it is that we're tasting, that's where we have our taste buds. And I always stress the fact that the thing that we think of as taste buds, those little bumps on our tongue, those aren't actually the taste buds. Taste buds occur in the grooves between those bumps. And I know that when I learned that, it kind of blew my mind because I spent my entire life thinking that the bumps were the buds. That made sense to me. But taste buds actually line the trenches in between each of those papillae. So that's our sort of new fact for the day. And so each of these little taste buds on the side are going to bind to chemicals that have dissolved in our saliva. They'll be receptive to those 5 different types of general tastes. And then they're going to send signals back to the brain through neurons, to sort of our sensory neurons, to let us know what we're tasting. And so they're going to travel through our thalamus, which is our routing station for the brain. That's basically what's sending stuff where it needs to go. It's going to go up through our insula and then to our primary gustatory area in the frontal lobe. So basically our primary taste center in the frontal lobes. And that's when we can sort of make those connections of what is it that we're tasting. We can put together our different ratios of those different tastes and combine it with other factors, maybe things like smell and texture, in order to get an understanding of what flavor it is that we're detecting. All right. So we want to talk very briefly about an interesting concept called sensory-specific satiety. And I can never say that word correctly, so if you know how to pronounce it right, absolutely correct me. But I look it up every single time, and then I forget how to pronounce it. But this is a behavioral pattern that means the more an individual eats a particular food item, the less appealing the food becomes. So this applies to many, many people. I am unfortunately not one of them. I could eat the same thing all the time and be perfectly fine with it, but that goes against this theory here. But this is the idea that you want to have a more diversified diet. You want to have lots of different kinds of nutrients. And if you eat the same thing all the time, then you're going to lose or sort of not get access to other different kinds of nutrients. So if you only ever make Kraft Dinner, then you're going to miss out on the things that are not included in that box of Kraft Dinner. And so we have sort of this built-in mechanism that encourages us to find other things appealing. For those of us who don't necessarily want to eat other things, I don't think it's actually the fact that we don't have a biological drive to try something else. I think it's more of a lack of motivation or laziness to make something else. But on the biological level, we are sort of more drawn to things that we haven't had a lot of or things that we haven't had recently. So if you think about what you've been having to eat for the last week, maybe you think of something that's completely different. Maybe there's a type of food that you haven't had in a couple of weeks, something that you're craving. It might be in a completely different category or include different balances of nutrients, different things that your body might be lacking, which would make it more appealing. So that's the basic idea behind this. We can also have some weird learning aspects along with our taste and what we choose to eat. The first of these is learned taste aversion. And this is when we specifically avoid foods that we've associated with getting sick. So I'll usually discuss learned taste aversion when it comes to, say you eat something or drink something, and if it makes you throw up, then your body is going to learn something very, very quickly about that particular food. I always use for myself the story of apple juice. I grew up in southern Ontario where in the summer you can hit sort of 35, 40 degrees Celsius, no problem. And as a child had left a juice box in the car, apple juice, and it sat there about 12 hours while we were at relatives. And then when we got back in the car, I was so excited because I had been waiting for juice all day, and I drank the whole thing at once, and it was probably really close to boiling at that point, and it made me sick. And I haven't intentionally had apple juice since. It's been probably 15 years, and I don't dislike apple juice. But anytime I think of it, I have that sort of initial, maybe not. And it's not because I don't like it, it's not because it's bad, but it's because I have a learned taste aversion that has made me really reluctant to have it again. You can have the same thing if you go to a restaurant that makes you sick, you might be reluctant to go back.
This is kind of just like a related story. I had a conversation the other day talking about throwing up a food and having it taste the same way coming up as it did going down, and then you just can't eat that food ever again. But
Yeah.
it's the same thing, right?
Yeah, and it's amazing how quickly we learn these things. When I teach classes on learning, sometimes when you're trying to teach your body something, it can take many, many, many exposures. Taste aversion is one of those things that is a single trial learning event where you do it once and your body is like, never again. And that makes sense evolutionarily, right? If you were out in the wild and you ate a strange new berry and then got sick, you probably shouldn't eat more of that. Mm-hmm.
It's funny how alcohol doesn't work that way.
Yeah, it's.
I have a comment about that. It kind of does and doesn't, but I find like that a lot of people say the first alcohol you got really, really drunk on, you can't drink anymore. So I think it kind of does, but it's just like the initial exposure.
It's especially strong for new things, which could explain why that is. If it's something that you've had a lot of, then not so much. Especially when you're young, they hit you really hard, which is probably why I went from having apple juice all the time to never again. But it's also for really distinct things as well. But if you get sick enough, you can have it, even for something that you've had a lot of. And that's actually sort of another little bit of a tangent. If you've ever known someone who's gone through chemotherapy, there's a really big problem where the people going through it really want to be feeling better, right? They're going through something that's awful, so they'll often choose to eat their comfort foods. But the problem is that chemotherapy makes them sick, and usually very sick, which will then lead to aversion for their favorite foods that they were eating to try and make themselves feel better. So it would come with this sort of compound where everything that they were trying to do to make themselves feel better, they were then losing their comfort foods, which is a huge problem. So what a lot of places will do now is have these really weird flavored ice creams or like popsicles, and they're going to be combinations that you have never experienced and will never see again, like something crazy strange like sweet potato lemon. And then the idea here is, especially because we form those associations a lot better with novel things that we've never had before, when they would then get sick after having these weird ice creams, their body would be tricked into going, oh, no, it's just the strange flavor, it's that new food. The stuff I had earlier doesn't matter. So there are ways that you can kind of circumvent it, but there's lots of moving factors here that get into it. But logically, there is sort of a benefit to it, and it can help us avoid things that have made us sick in the past, but it does cause problems nowadays in case you have sort of unrelated things. So even if you got sick and had even food poisoning, that could end up making you dislike something that you've always loved because it made you sick enough to override all of that earlier association. But yeah, a very, very interesting topic. We can also talk about learned taste preference. And so this is kind of the idea that some of the things that we have preference for are innate. So I think we've mentioned, and if not, I've mentioned it in my other class and I'm just overlapping, but when we're younger, we have this innate preference for sweet things. So when you're a child, things like candy and fruit and anything that has lots of sugar is really appealing. And there's actually, again, an evolutionary basis for this, because things that are sweet also tend to be high in calories, which is problematic right now in our society, where if you have easy access to a lot of things that have lots and lots of calories, then it can lead to obesity. But in nature, when you're young, the idea is that you want to grow and grow as quickly as you can with the best amount of energy that you can. So if you were exposed to sweet things, that's something that's probably high in calories, so that would be beneficial to keep eating when you're younger. And then as we get older, when it's less important to be bringing in massive amounts of energy to sort of fuel our growing bodies, then our taste preference for sweet things starts dropping off, which I always thought was ridiculous because I had a sweet tooth for my entire life. I would eat chocolate from sunup to sundown, and I was like, never, 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 never. And then mid-20s, I started being able to tolerate dark chocolate. And a little bit later on, now it's almost preferable to have the dark chocolate instead of milk chocolate, which just seems awful in my mind, but that's kind of what my body prefers now. So those preferences can change over time based on what's beneficial to have. That doesn't mean that all of our preferences are based on biology. There is some evidence that we tend to crave things that are sort of necessary for our body. So we'll have a momentary preference for a particular flavor. Maybe you're craving dill pickles, and it's because you're short on salt, or one of the other compounds that's in that particular food. So you might have a preference for something that you're deficient in. That's why a lot of people will say that you should listen to your body and sort of eat and drink the things that you want, because sometimes it's telling you that that's good or not. We also have learned preference that has nothing to do with what our body needs. And this is when we start sort of paying attention to the flavor or the food itself. And this can then form an association where we learn that, oh, I like this food, And our body learns that that food gives us that particular nutrient, and that's where some of our cravings can come. Because we don't naturally assume that, say, dill pickles are going to give us salt, but we've learned that if we're low on salt, having those would be beneficial. All right. Okay, and I think I covered all of that. And so then this diagram here, again, I like including some of the visuals from the textbook just in case you're a visual person, and otherwise the slides would get awfully boring. But the first thing that I wanted to mention is the fact that usually when you're taught about taste, they show you a map of the tongue. They call it a taste map, and they show you that the front of the tongue, the tip of the tongue, is where you taste sweet, and the back of the tongue is where you taste bitter, or whatever the arrangement is. And that's actually a really unfortunate bit of misinformation. So that was published originally when people were just starting to understand how our sense of taste worked. And as we've had more and more research, we've realized that all of our tongue has all of the different taste receptors. We just have them in different concentrations in different places. So if you look at the tip of the tongue, you have a few more sort of sweet receptors than other kinds of receptors, but not that the others are completely absent. So it's not like you can only taste sweet at the tip of your tongue and nothing else. And it's also not that the tip of your tongue is the only place you can taste sweet. So there are sweet receptors everywhere, and there are other receptors where we assume is associated with sweet. So that taste map, that colored in tongue that you usually see, is a little bit of misinformation. So we more have an equal balance. And then this diagram here is just showing us sort of our taste responses in our gustatory cortex. So this is that taste center in our brain showing lighting up based on different kinds of tastes. And so they've color-coded them there. And so it's that would be a more accurate map, would be mapping it into different locations in our brain than different locations on our tongue. Okay, and the next process is to get into the digestive process, but that's going to be a really big chunk of slides and sort of an in-depth process. So I think what I'll do to maintain our timing is I'll leave us here for today, and then we'll pick up with digestion in the next class. And then hopefully that'll keep us good on track.